The saber-toothed cat ranks among the most famous animals in prehistoric history. Yet most of what people think they know about it is wrong. It was not a tiger. It does not sit on the same branch of the cat family tree as lions or house cats. And when scientists finally sequenced its DNA, the results confirmed that this animal represents an entirely separate evolutionary experiment in what a large predator could become. This video focuses on that DNA, what it reveals about saber-toothed cats, and why the distance between them and modern felines runs far deeper than a pair of oversized teeth. If you value research-driven content like this, take a moment to like the video and subscribe before we continue. Confusion begins with the name saber-toothed tiger, a phrase that merges two animals that are not closely related at all. Tigers belong to the subfamily Pantherinae, alongside lions, leopards, and jaguars, whereas saber-toothed cats occupied a separate subfamily called Machaerodontinae, a lineage that vanished thousands of years ago. These branches diverged long before modern big cats appeared, and that split defines everything that followed. Picture the family tree this way. A house cat and a lion share a more recent common ancestor with each other than either one shares with a saber-toothed cat. Around 20 million years ago, and possibly as far back as 22.5 million, the Machaerodontinae line broke away from the lineage that produced every living feline today, which means saber-tooths had already been evolving independently for longer than the entire history of the human genus before the first human ever existed. That is a vast stretch of time. Two genera anchor most of the research, Smilodon and Homotherium, and each followed its own path. Smilodon, the heavily built cat most people picture, carried long stabbing canines and included a giant species called Smilodon populator that reached roughly 400 kilograms, about 880 pounds, outweighing even the largest Siberian tiger. Homotherium, often called the scimitar-toothed cat, developed shorter but still distinctive teeth and a body built along different lines, and DNA evidence later confirmed that these two predators differed not only from modern cats, but from each other as well. If this is your first time here, subscribe and turn on notifications so you do not miss what comes next. For decades, bones and teeth supplied nearly everything researchers knew about saber-toothed cats. Fossils reveal size, joint structure, muscle attachment points, and the mechanics of movement, yet they cannot expose cellular biology, genetic instructions, or precise evolutionary relationships. DNA provides that level of detail, and recovering it from animals extinct for more than 10,000 years presents serious technical challenges. Progress arrived step by step. In 2017, a research team that included Johanna Peumans from the University of Potsdam published the first mitochondrial genomes for both Smilodon and Homotherium, and mitochondrial DNA, passed down through maternal lines, offered enough information to clarify evolutionary placement with impressive accuracy. Those sequences confirmed that saber-toothed cats stood genetically apart from all modern felines, and the long-estimated 20 million year separation moved from fossil-based inference to molecular fact. October 2020 brought a deeper breakthrough. At the University of Copenhagen's Globe Institute, Ross Barnett and his colleagues sequenced the full nuclear genome of Homotherium latidens at 30 times coverage depth, meaning each section of the genome was read about 30 times to ensure reliability, and the result became one of the most complete ancient genomes ever assembled from an extinct species. That level of detail reshaped the discussion. Two findings from the 2020 study stand above the rest. In the University of Copenhagen press release, Ross Barnett described Homotherium as genetically very, very different from any cat living today, a statement grounded in direct comparison between its genome and those of modern big cats, where the differences proved deep and structural rather than minor variations of the kind seen between a cheetah and a leopard. Tens of millions of years of independent evolution left clear marks in the DNA. Michael Hofreiter from the University of Potsdam characterized Homotherium as a highly specialized predator, unlike any cat alive today, and the genome backed that assessment with concrete, genetic patterns. Specific adaptations pointed to high cardiovascular output, reinforced bone structure, and activity during daylight hours, which contrasts sharply with most modern big cats that concentrate their hunting at dawn and dusk. The genes align with endurance running and sustained aerobic performance, suggesting a predator that pursued prey over distance in full daylight rather than relying primarily on low-light ambush. Another discovery caught researchers off guard. Before extinction, Homotherium populations displayed high genetic diversity, a marker of large, widespread and healthy populations where harmful mutations were diluted instead of amplified through inbreeding. And that evidence runs counter to earlier theories claiming genetic deterioration drove saber-toothed cats toward extinction. The DNA does not support that scenario. Although the 2020 genome focused on Homotherium, Smilodon contributes its own crucial evidence through the fossil record. Jaw mechanics stand out immediately because Smilodon could open its mouth to roughly 120 degrees compared with about 65 degrees in a modern lion, and that extreme gape required a different skull design, altered musculature, and a killing technique unlike anything living cats employ. This was a distinct mechanical system. Los Angeles provides the richest source of Smilodon fossils, and the La Brea tar pits have produced more specimens than anywhere else in the world. Many of those skeletons show healed fractures and damaged joints that recovered rather than causing death, and an injured apex predator that cannot hunt alone would require support from others, which points towards some level of social cooperation. The genome has not resolved Smilodon's social structure, yet the fossil evidence leans in that direction, and nothing in the genetic data contradicts it. Debate continues over how Smilodon used its elongated canines. One interpretation argues for slashing soft tissue in the throat to induce rapid blood loss. Another favors deep stabbing into vital areas, and when that discussion is set alongside the Homotherium genome, which indicates endurance running and daylight activity, the contrast becomes clear. These genera did not represent minor variations on a single predatory model, but separate solutions to life at the top of the food chain. Around 10,000 years ago, near the end of the Pleistocene epoch, Smilodon disappeared along with many other large mammals. Climate shifts at the close of the last ice age altered habitats. Large prey species such as mammoths and giant ground sloths declined, and expanding human populations entered the Americas, and most researchers point to some combination of those pressures, with prey collapse playing a central role. An apex predator specialized for megafauna faces a direct crisis when that megafauna vanishes. De-extinction often enters public conversation, yet the genetic distance highlighted earlier creates a major obstacle. Roughly 20 million years separate Machairodontinae from every living cat, and without a closely related species to serve as a surrogate mother, cloning or genetic reconstruction cannot produce a true saber-toothed cat, because lions and tigers differ too greatly at the molecular level to carry such an embryo successfully. Current reproductive technology cannot bridge that divide. Work continues nonetheless. As of 2024, researchers are attempting to extract usable nuclear DNA from Smilodon fatalis specimens at the La Brea tar pits. Although the asphalt that preserved so many fossils complicates recovery of genetic material, and the most complete ancient genomes so far have come from Homotherium remains found in the Yukon and Siberia, where cold conditions preserved DNA far more effectively than tar ever could. Saber-tooth DNA records more than the story of a single extinct predator. Over more than 20 million years of independent evolution, Machairodontidae developed answers that diverged sharply from those of the modern cat family, which refined flexible bodies, retractable claws, low-light vision, and ambush tactics across dozens of species on every continent except Antarctica. Different blueprints produced different results. Extreme jaw mechanics built around a killing technique absent from today's world. Cardiovascular systems in at least one genus tuned for sustained endurance. Body masses that surpassed the largest modern big cats, and fossil evidence hinting at social complexity all point to a lineage that charted its own course, and genomic sequencing now allows researchers to see those differences written directly into the biological instructions that shaped the animal. Bones suggested divergence. DNA confirms it. When Ross Barnett described Homotherium as genetically very, very different from any living cat, he was summarizing more than a comparison of sequences. He was pointing to a separation that began around 20 million years ago and ended with an entire subfamily leaving no descendants behind. And the genomes recovered from permafrost now stand as detailed records of how those animals were built and how far removed they are from anything alive today. If research-driven deep dives like this are why you are here, like the video, leave a comment with the topic you want covered next and subscribe so you receive every new release. Every claim in this script comes from sourced research, and that standard guides the entire channel. Turn on notifications, and I will see you in the next one.